Assalamualaikum, I am Khalisa Husna binti Azmi, and today I will be presenting about my final year project, which is a review on the effect of captivity on reproductive success of the endangered wildlife. Endangered animals are brought into captivity for many reasons: conservation, research, agriculture, and the exotic pet trade. Various in situ and ex situ conservation initiatives are being implemented all over the country to conserve and prevent these species from becoming extinct soon. Despite the effort, their natural habitats are still being destroyed due to lack of adequate habitats, heavy deforestation, and land use conversion. Although the physical needs of animals are met in captivity, the condition of confinement and exposure to humans can result in psychological stress. This stress could reduce their productivity as well as their reproductive performance, since Fisher once reported that not all animals could survive well in captivity back then in 2019. Therefore, there should be more discussions on this topic in particular, which leads to my objectives. As you can see, it delineates to 4 parts. First, to discuss the fundamentals of stress due to captivity. Second is to explain the detailed ways in which stress influence the reproductive success of the threatened wildlife. Next is to address the issues in captivity that cause stress. Last but not least is to highlight the ways to enhance the reproductive performance of the captive endangered animals. Apparently, you can write a thesis without knowing the gaps in research papers that came before yours. As you can see, my first question is what is meant by chronic captivity stress in endangered wildlife? What occurs during the stress mechanism? What is the relationship between captive stress and their reproductive efficiency? What issues that cause stress to threatened wildlife in captivity, and how to enrich their captive environment so that the animals can reproduce successfully? So, to answer the research questions that I stated just now, here we go. What is chronic stress? In 2016, Cohen and his colleagues reported that chronic or bad stress occurs when external demands exceed an organism's adaptive capacity, causing psychological and biochemical changes that can lead to disease. This chronic stress is condition caused by a set of conserved physiological response known as stress response, and this stress response is stated to occur when events are viewed as dangerous, regardless of whether the animal is experiencing physical injury or psychological struggle. It is later proven by Ladenpera and Oyelai that mated captive-born females could not produce offspring as well as wild caught females due to reproductive problems such as post-copulation problems and inability to conceive or maintain pregnancy. How does it happen? As you can see in figure 1 I made, it is all started when the stress response triggered by any physical or psychological stresses, like how the researchers have been mentioning before, which then activates the sympathetic adrenal medullary axis, which is called as SAM axis, and hypothalamus pituitary adrenocortical axis, which is called HPA axis. A little bit about these two. The SAM axis mediates acute stress response, while the HPA axis mediates both acute and chronic responses. Since I've been talking about chronic stress today, therefore I'll be focusing on HPA only today. So HPA induces the hypothalamus to release the corticotropin releasing hormones CRH and vasopressin VP, which in turn causes the pituitary gland to release adrenocorticotropic hormone ACTH, and this ACTH causes the adrenal gland to release glucocorticoids. When the hypothalamus and anterior pituitary sense high GC levels, they use VP, CRH, and ACTH to stop the adrenal gland from making more GCs. This somehow stops the stress response. I have been mentioning a lot about GC, right? Yep. It is because one of the most common variables to measure when assessing the stress of captivity was GC concentration. The degree of GC effects is actually determined by how much and how long GC is produced and how much GC metabolite are broken down. GC hormone secretion could inhibit reproductive function by acting on animal cell, which it prevents basic cell function from happening by causes the proteins to fold in a wrong way. As a result, cellular respiration, ATP production, and even testosterone production can happen. While in the ovary, it prevents the expression of cyclooxygenase 2 and lipoxygenase, which then inhibit the production of PGE2, which is essential for ovulation. And in testes, GCs could prevent conversion of testosterone to dihydrotestosterone by inhibiting the 5-alpha reductase. You can see in details in my figure 2. So, what exactly causes these captive animals to feel stress? First, physical enclosure issues. Many researchers have been highlighting how the size and the design of the enclosure is one of the most common causes of restricted behavior in captive animals. For example, it was reported back then in 2018 by Schultz and Young, the general activities of the spectacled bears and Indian leopards increased when they added climbing structures in their enclosure. And second is social structure issues. Has been shown in various species that investing in the creation of long-term stable connections improves the animal's quality of life and reduce stress levels. And for example, the Asiatic golden cat had difficulties reproducing in captivity due to social isolation and lack of early bonding with conspecifics. It was by Chatterjee in 2018. Next, we have captive breeding issues. Captive breeding efforts, on the other hand, frequently result in injury and death due to animals' aggressiveness and stress. Inbreeding is an example of problem in captivity that results in poor health and mortality, reported by Arumugam and Anavi in 2019. And finally, we have nutritional issues. Each animal species has evolved a specific diet based on the food available in its native environment, how it is provided, and presence of competing species. And here we go, ways to enhance the reproductive performance in captive environments. First is by enriching the enclosure design as well as the environment. Captive settings should be spacious enough to allow for a full scope of locomotor activities such as walking, climbing, swimming, burrowing depending on the species. And next is by using the assisted reproduction technologies such as in vitro fertilization, IVF, artificial insemination, multiple ovulation and embryo transfer called MOET. Overall, I could say that the endangered wildlife adapts to captivity in largely species-specific manner. And I would like to suggest, in order to boost their reproductive success, maybe we can change the time of the year they are captured, enhancing and enriching their habitats and perhaps employing pharmaceuticals, who knows. After all, captive wildlife, especially the endangered ones, must be able to reproduce effectively to maintain its population. So here are some of my references, and thank you for your attention and have a good day.